Hello, anatomists and physiologists. This is another video in our immune system series. We're finishing up talking about adaptive defenses. In the last video, we talked about B cells, the humoral response. Now let's shift our focus to T cells. There's two major types of T cells. There's CD4 cells, which are called helper T cells, and there's CD8 cells. These are called cytotoxic T cells. So let me label these for us. So CD4 T cells are also called helper T cells. Or TH. CD8 T cells are also called cytotoxic T cells or TC. T cells are kind of like in their own world for their life. B cells, I think they have more street smarts. They know an antigen when they see it and they can make antibodies for it. But T cells, when they're immature, they're considered naive until an antigen presenting cell shows it to them. But like it needs to be a self cell showing it to the T cell. It's like T cells can't trust what they're seeing is really an antigen unless that information comes from a self cell. You know what I mean? Okay. So first we'll talk about helper T cells, the CD4 guys. Remember that the self protein on the outside of cells is called MHC. So remember that if we have a self, we have the self protein complex, that's called MHC. There's two major types of MHC. There's MHC1 and MHC2. But the MHC protein complex, kind of like if we're to zoom in on the MHC protein complex, it kind of looks like, well, there's like a little pocket in there. So if I were to erase this MHC label, here's my MHC complex, and there's like a little pocket there. That's the MHC. There's a little pocket in there, and that little pocket is where we can find an antigen. Oops. That's the thing that the T cell wants to recognize. So again, there's two types of MHC. There's MHC1 and MHC2. So we have MHC1. That's found on like our normal body cells, except for red blood cells. So normal body cells. But also dendritic cells too. And we also have MHC too. Right. Okay. And MHC2, those ones are the ones that are found on antigen presenting cells, APCs. Dendritic cells, macrophages, B cells. The structure of MHC is really cool, like I said, because there's that little pocket where the antigen hangs out. And so in order to present to the T cell what's going on, that antigen and the MHC complex must both be present. But there's two things that can happen. In cells that have MHC1, that antigen can be a normal like self protein. And if that antigen is like a normal self protein, when we go to introduce that MHC complex with that little protein there, the T cell's like, oh, you're me. That's fine. Okay, I'm just going to ignore you. But the other thing that can happen is that antigen is not ours. So let me show you what that would look like. So if we have a self cell, and this antigen is not ours, it's foreign, that means that antigen, in order to get into that MHC complex, had to come from inside of the cell. And so this antigen would be what we call endogenous. That word endogenous means from within. That means that the cell, that self cell, is infected or its machinery isn't working properly. So what's going to be here is infected proteins or irregularly shaped proteins because whatever's going on inside the self cell is wrong. And that alerts the T cell to be like, oh no. So that way the T cell can do something about it. And that's for MHC1. So that antigen can be endogenous. So MHC1. This happens when we're talking to, so that MHC1 situation happens when we're talking to CD8 cells. So that happens when we're talking to those cytotoxic T cells, if that protein then is endogenous, that antigen protein rather. If the opposite is true, if it's an MHC2, so here's our self cell, here's our MHC complex with their little pocket, and here's our antigen. So we have MHC2, here's our antigen. If that is an MHC2, that antigen, because of the structure of how it works, that antigen comes from the outside of the cell. So that means that this cell, this self cell, isn't infected with that antigen. It comes from our antibody presenting cells. So MHC2, this is usually an this is usually an APC, antibody presenting cell, a dendritic cell, a macrophage, a B cell. What happens with APCs is that they engulf that antigen, they smoosh it up into pieces, and then they stick that antigen right on that MHC2 complex. So that class 2 MHC is what the CD4, the helper T cells, recognize. So that little MHC has a little pocket where the antigen lives, and that slides right into that helper T cell. But notice that while those T cells get activated, we still form memory cells, we still form effector cells that are going to make all of the actual immunity part happen. So those effector cells that are going to get rid of or help activate, in the case of helper T cells, all of the things that we need to get rid of. So how are T cells actually activated? There's 3 major steps here. The first step of T cell activation and differentiation is just what we discussed. The antigen is presented to the T cell and in that little MHC complex pocket on the antigen presenting cell or whatever cell. Then this idea of co-stimulation happens. It's when the T cell must bind to something else on the antigen presenting cell. So T cell must bind both to the MHC with antigen and the co-stimulation molecule. In that way, if this doesn't happen, if that co-stimulation step doesn't happen, the T cell can't carry out its job. It's like the secret handshake that the T cell and the antigen presenting cell must do to make sure they're on the same page. So without co-stimulation, stuff's not happening. So now that the antigen binding is done, the co-stimulation is done, now the cell will divide, proliferation. Proliferation happens, and this is because there's a bunch of chemical messengers or cytokines. So this is due to cytokines. There's a big old list in your book, so I'll let you read about that. But those cytokines are released by antigen-presenting cells. And they make T cells divide a lot. Remember, we still produce memory cells in the process, so we're still on the right track. When we produce those effector T cells, they do their thing, but then they don't live that long. It's only like 30 days or so. Because T cells, they're a little bit extra. If there's too much division of T cells happening, that can lead to errors in cell division, and that can lead to cancers. But we still have those memory cells as backup, so we just don't want the T cells to live that long. Just long enough to do their job. So then the question becomes, what do T cells actually do? This slide is about helper T cells. Helper T cells, they help. They don't actually directly kill cells, but what they can do is activate B and T cells and cause them to proliferate. They can also secrete cytokines to call on other immune cells. And when our helper T cells, our CD4 cells, don't work or they're destroyed, this is devastating to the immune system. And that is a hallmark of the disease AIDS. Okay, so there's two different situations going on here. There's the green panel and the yellow panel. So to activate B cells, what happens is there's MH2 receptors on B cells, and then our helper T cells. Let me highlight here. So we have our B cell is being activated here. So we have MHC2 receptors, MHC2. We have our helper T cell binding, and this causes cytokines to be released. And ultimately, it makes B cells divide a lot faster, and it also signals them to make lots of antibodies. So they release stimulatory signals to complete B cell activation, which leads to a bunch of B cell proliferation, which leads to lots of antibodies. Yay. Then the yellow panel is about CD8 cells, those cytotoxic T cells. Cytotoxic T cells need a little help from the helper T cells to be activated because the helper T cells encourage the dendritic cell, the antigen presenting cell, to show its co-stimulatory molecule. So a lot of science words, let's talk about it. So first what happens is that the helper T cell binds the dendritic cell. Remember that dendritic cells are antigen presenting cells. So they have that MHC2 protein. They have that foreign antigen there in the middle. And the T cell recognizes it. So that T cell recognizes it. This is the helper T cell. The helper T cell sends some signaling molecules in order for the co-stimulation bit to happen. Remember, if co-stimulation doesn't happen, nothing happens. So the helper T cell encourages the dendritic cell to release its co-stimulatory molecule. The co-stimulatory molecule is this guy. That's the double handshake, the secret handshake that we need to do. So we not only need to bind to the cytotoxic T cell, but we also need to have the co-stimulatory molecule. So it's a double round of checking in order for the T cell to become activated. There's another kind of CD4 helper T cell not listed here, which is a regulatory one. And that slows down the immune system, which is important for controlling like the actions of autoimmune diseases and things. Okay, so that's helper T cells. This next slide is about cytotoxic or CD8 cytotoxic T cells, or TC. Lots of different names here. Cytotoxic T cells are the ones that can actually kill other cells. They hang out in the blood and the lymph and all over, just watching and waiting to be activated. They can bind to class 1 MHC antigens, so MHC1s. They bind to class 1 MHC antigens, and they notice the stuff that needs to be destroyed, the infected cells, the abnormal cells. Because remember that or that antigen sitting in the MHC1 pocket is from infected or abnormal cells. So that's important. So its machinery is thrown off, that cell is infected, so it's making some weird stuff. Either way, the CD8 cells are like, I need to just destroy this cell. And then this prompts a release of enzymes, two major enzymes here, perforin and granuzyme, which ultimately, through a series of steps, make the cell undergo apoptosis, programmed cell death, pushing the big red self-destruct button on its own self, prompted by the CD8 cytotoxic T cell. This figure is showing you a summary of primary immune response. I really like it, and I think you should look at it. It's figure 21.1 in your textbook, and there's even an animation that goes with it. So this is the first page, and this is the second part of that picture. I just couldn't fit it all on the same slide. I really think that you should go take a peek at it and look at it.